Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
The science of beer. Microbiology, raw materials, and brewing process explained. Table of contents. Introduction. Beer production history. Beer production raw materials ingredients. Beer production process, malting and brewing. Malting process. Brewing process. Beer production byproducts and waste recycling. Beer and health. Beer production and environmental effects. Conclusion. Introduction. Beer is a widely consumed alcoholic beverage worldwide. It ranks as the third most popular drink globally, following water and tea, due to its unique taste, aroma, and appearance. The taste, color, and smell of beer are determined by its chemical composition. Beer consists of approximately 90% water, containing mineral ions, while the remaining portion comprises organic compounds such as carbohydrates and aromatic molecules. Often referred to as liquid bread, beer is rich in micronutrients, including magnesium, vitamin B, biotin, potassium, and phosphorus. It also contains phenolic acids like vanillic acid and 4-hydroxyphenyl acetic acid. Additionally, beer includes 8-prenylnaringenin, a phytoestrogen derived from hops, contributing to its bitter taste and distinct aroma. The production of beer involves brewing and fermenting starch from grains such as malted wheat, rice, maize, and oats. Home brewing is also possible using a home brewer. During fermentation, yeast converts starchy sugars into ethanol, resulting in alcohol production and natural carbonation in the final product. Most modern beers incorporate hops as an additive, serving as a stabilizer and preservative while imparting a characteristic bitter flavor. The brewing and fermentation process involves a minimum of 10 steps, transforming malted barley or other grains into the final beer product. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Beer production history. Beer is one of the oldest known alcoholic beverages, with evidence supporting its ancient origins. The simplicity of the beer-making process contributed to its early invention and widespread popularity. Since any starch-containing substance can undergo alcoholic fermentation, many cultures quickly developed and adopted beer as a sweet liquid. The earliest archaeological evidence of beer fermentation dates back 13,000 years, found in beer residues used by semi-nomadic groups in the Raqefet Cave of the Carmel Mountains, where it played a role in ritual feasting in Israeli culture. In Sumerian culture, the famous hymn, the Hymn of Ninkasi, functioned both as a prayer and a method to preserve the beer recipe among the few literate individuals. Brewing techniques reached European territories around 3000 BC, where primary fermentation was commonly performed in households. European beers were often flavored with fruits, herbs, and spices to enhance their taste. With the Industrial Revolution, beer production expanded significantly, leading to widespread mass production worldwide. Beer production. Raw materials. Ingredients. Any type of beer requires 4 essential raw materials for the manufacturing process: malt, water, yeast, and hops. These ingredients influence the taste, aroma, and color of the final beer product. Water, major raw material. Water makes up approximately 90% of beer composition. Modern technologies allow precise modulation of salt and mineral ion concentrations. While water itself is flavorless, bicarbonate ions contribute to the taste of the final beer. The water used in brewing must be pure, potable, and pathogen-free, which is ensured through chemical and microbial analysis. The mineral composition of brewing water varies by region, influencing the type of beer produced. For example, Guinness is brewed with Germany's hard water, which has high salt content, while Pilsner is brewed with low salt water from the Czech Republic. Starch source: grain malt. The primary starch source comes from the malting of germinated seeds. Malting involves soaking grains, such as barley, wheat, or rice, in water to allow germination, followed by drying in a kiln. During malting, enzymes break down starch into fermentable sugars, making the grain malt a key determinant of beer flavor and strength. Different types of malted grains produce beers with unique flavors. Wheat malt has a higher protein content, producing a foamy head and a fuller taste. Corn malt results in a lighter beer with reduced haziness, enhanced flavors, and a more neutral sweetness. Hops, additive. Hops flowers are widely used in modern beer production to balance the sweetness of malt with bitterness and act as a natural preservative. Essential oils in hops give beer its distinctive aroma. Hops contain over 200 compounds, including resins, polyphenols, and flavoring agents, which influence the beer's taste. Only cones from the pistillate hop plant are used for brewing. Alpha acids and their derivatives provide bitterness and enhance preservation. Resins in hops improve beer's physiological digestibility, foam stability, and antibiotic properties, preventing the growth of undesirable microorganisms. The presence of polyphenols like xanthocumol contributes to beer quality, with xanthocumol exhibiting anti-carcinogenic properties. Yeast, fermentation agent. Yeast is the microorganism responsible for fermentation, converting sugar from malt, wort, into alcohol and carbon dioxide. The preferred yeast belongs to the Saccharomycetaceae family, commonly known as brewer's yeast. Two dominant fermentation techniques are widely used in beer production. Top fermentation. Yeast extract is layered on the surface of the malt. Saccharomyces cerevisiae is the preferred yeast strain. Fermentation occurs at 15 to 26 degrees Celsius. Bottom fermentation. Yeast extract settles at the bottom of the beer must. Saccharomyces pastorianus is the primary yeast strain used. Fermentation occurs at 8 to 14 degrees Celsius. Beer production process: malting and brewing. The primary goal of beer production is to convert grain starch from germinated seeds into fermentable sugars, extract these sugars using water, and then ferment them with yeast to produce an alcoholic, carbonated beverage. The beer-making process consists of multiple steps, including malting, preparing the grain by allowing it to germinate and then drying it; milling, crushing the malted grains to expose the starches; mashing, mixing the crushed grains with warm water to convert starch into fermentable sugars; extract separation, separating the liquid wort, sugar-rich extract, from the solid grain residues; hop addition and boiling, adding hops to the wort and boiling it to enhance flavor, bitterness, and preservation; removal of hops and precipitation, filtering out hops and allowing unwanted proteins to settle; cooling and aeration, lowering the wort's temperature and introducing oxygen to aid yeast growth; fermentation, adding yeast to convert sugars into alcohol and carbon dioxide; separation of yeast from young beer, removing excess yeast from the newly fermented beer; aging and maturation, allowing the beer to develop its full flavor and carbonation; packaging, bottling, canning, or kegging the final product for distribution and consumption. Malting process. Malting is referred to as the artificially induced germination of crop and involves several key steps: steeping, aeration, germination, drying, and curing of malt. The primary goal of malting is to promote enzymatic activity that facilitates the breakdown of complex starches and proteins, making them suitable for fermentation. the ideal protein content for conventional beer, is estimated to be 11 to 11.5%, commonly found in barley. Barley, with its high protein content, can contribute to colloidal instability, affecting beer clarity and quality. Heterogeneous malting, which involves using grains of varying sizes, can lead to inconsistencies in germination rates, impacting final beer quality. Smaller grains tend to germinate faster and contain higher protein levels compared to larger grains. Steeping. The process begins by immersing barley grains in water to initiate hydration. Barley is typically harvested at 12% moisture content and then steeped in water at 12 to 15 degrees Celsius for 40 to 50 hours. During steeping, the grains absorb water, leading to a 25% increase in volume and a rise in moisture content to 45%. A white root sheath called chit emerges through the husk, marking the start of germination. The steeped barley is drained and subjected to air rests, promoting oxygenation before germination. Germination. Germination requires a controlled environment with sufficient heat, oxygen, and humidity. The optimal temperature for germination is between 14 to 18 degrees Celsius. Traditionally, the steeped grains are kept in heaps for 24 hours before being spread on a floor for continued germination. During this phase, key enzymes such as alpha and beta amylases and proteases are formed. These enzymes help break down complex glucans and insoluble proteins into soluble glucose and amino acids, facilitating fermentation. As germination progresses, enzymatic activity intensifies, further converting starches into fermentable sugars. Kilning of malt. Kilning is the drying process that stops germination, removes excess moisture, and develops the color and aroma of the malt. This step arrests 30 to 40% of enzyme activity while preserving essential compounds. Kilning is divided into two phases. Withering. The water content is reduced from 45% to 10% at low temperatures. Curing. Conducted at 80 to 105 degrees Celsius, this stage enhances malt color and aroma. Higher temperatures produce darker malt by reducing dimethyl sulfite, a compound responsible for beer aroma. If the temperature remains below 40 degrees Celsius, with water content above 20%, the grain remains in the growing phase, where enzymatic degradation continues. At 40 to 70 degrees Celsius, the process enters the enzymatic phase, maximizing enzymatic activity and starch breakdown. As water content decreases, degradation slows, halting embryo growth and allowing the accumulation of breakdown products. During kilning, radicals must be removed to prevent excess water absorption, which can lead to increased bitterness and dark coloring. Malting adjunct. Malting adjuncts are additional starch or sugar-based materials used to dilute excess proteins in barley. One common adjunct is dextrose sugar syrup, which helps regulate fermentation efficiency and contributes to beer consistency. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Brewing process. The brewing process involves multiple steps, each contributing to the final quality of the beer. First, milling. Milling is the process of crushing dried barley grains between rollers to produce a coarse powder known as grist. The quality of milling significantly impacts subsequent stages like mashing and lautering, ultimately influencing the beer's final characteristics. Finely milled malt increases the surface area, enhancing enzymatic reactions and improving ingredient dissolution. Second, mashing. The grist is mixed with warm water and maintained at 62 to 70 degrees Celsius to facilitate enzymatic breakdown. This process hydrolyzes starch into simple sugars through amylase enzymes and breaks down proteins into smaller peptides and amino acids. The effectiveness of enzymatic hydrolysis depends on pH and temperature. Key processes in mashing. Amylolysis. The breakdown of starch occurs in 3 phases. Gelatinization. Starch absorbs water and swells at 60 degrees Celsius in the presence of amylase. Liquefaction. Amylases digest gelatinized starch, breaking down amylopectin and amylose. Saccharification. Dextrins are further reduced to maltose. Alpha amylase is most active at 57 to 65 degrees Celsius, while beta amylase functions optimally at 70 to 75 degrees Celsius. Proteolysis. Proteins are broken down into smaller peptides and amino acids by endopeptidases, which cleave proteins internally, increasing soluble nitrogen. Exopeptidases, which break down protein chains at the ends, forming free amino acids. Protein degradation influences beer's flavor and foam stability. Cytolysis. Hemicellulose in the cell wall degrades, increasing malt viscosity. This occurs below 50 degrees Celsius, but the breakdown slows as temperature rises. Drittes wort. Preparation. The mash is transferred to a mash tun, where water addition enhances enzymatic reactions. In the high-short mashing procedure, the mash is maintained at 60 to 65 degrees Celsius for 30 to 45 minutes before being heated gradually to 72 degrees Celsius. Once iodine normality is achieved, the liquid extracted is called wort, which contains approximately 10% sugars, along with carbohydrates, proteins, and other organic molecules. Viertes. Lautering. Wort separation. Lautering separates the wort from solid grist in a specialized container called a lauter tun. The grain husk serves as a natural filter. Initially, wort is drained with an extract content of 16 to 20%. The remaining grist is flushed with hot water multiple times, known as last runnings, with an extract content of 0.5 to 1%. Temperature regulation is critical. Higher temperatures reduce viscosity and accelerate lautering. However, temperatures above 80 degrees Celsius deactivate alpha amylase, halting starch breakdown. Fünftes wort, boiling and hops addition. The separated wort is transferred to a kettle for boiling, an essential step to halt enzyme activity. Extract bitterness from hops. Evaporate excess water and adjust extract content. Enhance flavors, color formation, and protein precipitation, flocculation. Sterilize the wort. Liquid sugar adjuncts, such as corn syrup and dextrose, are often added. After boiling, hot trub, precipitated proteins and hop particles, is removed. To prevent contamination, the wort is rapidly cooled before fermentation. 6. Fermentation by yeast. Yeast strains used for fermentation include Saccharomyces, Pastorianus, and Saccharomyces, Carlsbergensis for bottom fermentation, Saccharomyces cerevisiae for top fermentation. Process. Yeast inoculum, 0.3 kilograms per hectoliter of wort, is treated with tartaric and phosphoric acids to lower pH and prevent bacterial contamination. Fermentation occurs at 3 to 4 degrees Celsius for 14 days. Yeast converts sugars into alcohol and carbon dioxide, with minor byproducts like acetic acid and glycerol. In closed fermenters, liberated CO2 is collected for carbonation. By day 5, maximum CO2 is produced, and by days 7 to 9, yeast cells flocculate and become inactive. The final beer typically contains 3.5 to 5% alcohol. 7. Maturation and carbonation. The young green beer is stored at 0 degrees Celsius for several weeks to months. This period allows yeast, resins, and proteins to precipitate, clarifying the beer. Esters and other compounds develop, enhancing aroma and taste. After aging, the beer undergoes forced carbonation using CO2. 8. Pasteurization, filtration, and packaging. Pasteurization at 60 degrees Celsius eliminates residual yeast and spoilage microbes, extending shelf life. Filtration removes any remaining particles, ensuring clarity. Packaging involves filling kegs, bottles, and barrels under sterile conditions at 60 to 65 degrees Celsius. Removing air from containers prevents oxidation, preserving beer freshness. Byproducts and waste recycling. Brewing generates several byproducts that can be repurposed. Rootlets discarded during malting serve as animal feed. Spent hops are used as fertilizer. Residual yeast is rich in vitamin B and utilized in pharmaceuticals and food additives. Recycled bottles and cans contribute to sustainable production. Byproducts and waste recycling in beer production. Beer manufacturing generates several byproducts and waste materials that can be repurposed in various industries. Grain rootlets. Discarded rootlets from the malting process can be collected and used as animal feed. Spent hops. The hops left after wort extraction serve as a natural fertilizer for crops. Residual yeast. Yeast recovered from the brewing process is a rich source of vitamin B and can be utilized in pharmaceutical industries for producing medications, vitamin supplements, or as a food additive. Recyclable packaging. Used beer bottles and cans are regularly collected and recycled to minimize waste. Beer and health. Studies present varying perspectives on beer consumption as numerous factors influence its health effects. The impact of beer on health depends on variables such as region, lifestyle, and socioeconomic status. Excessive beer consumption can lead to alcoholism, which has been linked to a reduction in life expectancy by approximately 10 years, making it the third leading cause of premature death in the United States. However, moderate beer consumption has not been associated with increased mortality or abdominal obesity. In men, moderate intake has been linked to a lower risk of diabetes. The condition commonly referred to as beer belly is primarily caused by factors such as poor muscle tone, a sedentary lifestyle, and overeating rather than beer consumption itself. Ultimately, the health effects of beer depend on multiple factors, including the quality of the beer consumed and an individual's overall health status. Beer production and environmental effects. As global beer consumption continues to rise, achieving sustainable development has become a key priority for both manufacturing industries and consumers. The regulation of raw materials used in beer production is carefully managed to prevent the overuse of natural resources. Various factors contribute to the environmental impact of beer manufacturing, including the materials used for packaging, such as beer cans and bottles, agricultural inputs like fertilizers and pesticides, and transportation, which involves petroleum fuels and the release of toxic gases from vehicles. Additionally, the thermal energy generated during the malting and brewing process further challenges sustainability efforts. Addressing these environmental concerns through improvements in production methods and resource management is essential for fostering more eco-friendly practices within the industry. Conclusion. Beer, the third most consumed beverage globally, is often referred to as liquid bread due to its popularity. It has a distinct combination of sweetness, bitterness, and aroma. Comprising 93% water, beer also contains alcohol, essential oils, vitamins, and carbohydrates. Standard beer typically has an alcohol content of 4.5 to 5%, in accordance with industry regulations. The brewing process involves several carefully controlled steps, including steeping, lautering, and fermentation, all maintained at specific temperatures and pH levels. Beer is primarily made from malted grains such as barley, oats, rice, and wheat. each imparting unique flavors to different beer varieties. The type of water used in brewing also affects the final taste. Hard water is used for Guinness beer, while soft water is used for Pilsner beer. Although beer does not offer significant health benefits, moderate consumption has been linked to a reduced risk of diabetes in some men. To promote environmental sustainability, both the beer industry and consumers must actively participate in efforts to minimize the ecological impact of beer production. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!